So our next guest coming up here, we're at what, 3:40. Man, I'm trying to stay on time, Lee. Oxana is our next guest, Senior Vice President, Research of Dyne Therapeutics. I would love to pronounce her last name, but unfortunately on my copy, half of it got chopped off. So I apologize, doggone it. There she is. Hello, Oxana. How are you doing?
Hello, Tim. Very happy to meet you today. Delighted to be here. I'm delighted to participate in Giving Tuesday's Bellathon.
This is a blast for me, meeting so many different people. You are one of those new faces I have not met. So, hello, nice to meet you. Thank you for spending the time today, this afternoon, and joining us on this event. Also, thank you to Dyne for being a sponsor. Huge help, huge support. We've talked with Molly White already earlier today, and she's been on in the past. So we've had good conversations there about what Dyne is doing. And I guess that's where I'm going to start, right? I'm going to start a little about you and what you do at Dyne, and kind of introduce yourself to our audience, those that might not know who you are.
Happy to do so. I am a Senior Vice President and Head of Research at Dyne. As you know, Dyne is a young company, about 3-year-old biotech that is focused really on developing, transforming therapies for people living with serious muscle diseases, FSHD being really one of them. Just a little bit about myself. I am a scientist. My whole scientific career was really driven by a strong interest in human genetic diseases. I can tell you this has been just a fascinating way to understand how inherited mutation leads to the development of symptoms. Why in some people the disease manifests early in life, why sometimes it's late in life. What are those events that affect normal tissue, and within tissue its composition of cells and molecular events? And really the interest was driven by the desire to use this knowledge then to understand how can we treat the disease and be kind of focused on actual molecular defect, molecular cause that is initiating this cascade of events. And so I can tell you that I actually got my training in muscle biology after getting PhD in genetics. And I was lucky enough to join the lab of Kevin Campbell, who is at the University of Iowa, a renowned leader in muscle research. And if you think about years, it was actually productive work that was done beforehand. And Luke Uncle discovered dystrophin as an important protein for Duchenne. And in Kevin Campbell's lab, I was lucky to participate in understanding how dystrophin functions in muscle cell, what is the complex that allows it to attach to the muscle membrane. And through this understanding, we were able to actually begin to unravel molecular pathogenesis of other form of disease. And so here we are today. Many years back, a lot of good research is done in FSHD. And some years then spent at Genzyme, developing, again, therapies for multiple genetic human diseases, but always staying in the field of rare diseases. Not necessarily in muscle, but I also contributed to kidney, rare diseases, skeletal, and then back to the muscle disease. And then, interestingly enough, I joined Dyne, which was for me a very natural and easy decision because I met the Dyne team at the time that the company was starting, developing very interesting new technology, and really meeting this patient-centric young company with dedicated team that is really had a single mission in mind. It's really going after muscle diseases, serving patients with innovative therapy with a new approach.
That you kind of led into the next question. So great segue there. It's what is Dyne's strategy specifically with FSHD therapy? You know, your job or what you think is the pathway to a therapy.
Right. And again, sort of a little bit about DINE, and I will explain then essential pieces. So what is, how do we approach FSHD specifically and other muscle diseases? First, we understand in depth FSHD pathobiology. A lot of work was done by phenomenal scientists. Many of those are talking to you today as well. So a lot is done ahead of us. A lot is done not only in understanding the disease, in talking to patients, you know, talking to FSHD people gives us this unique view of experiences, because what actually I learned over years, and many researchers in rare disease field, we share the same theme, is people afflicted with the disease are best expert in their own disease. So it's, we not only, we are talking to you guys because we are interested to meet you, because you know so much, because you're typically very involved, motivated people, but also you possess a unique knowledge that is important to us. It's really a collaborative work. And so what is specifically DINE's approach? So we are capitalizing on the understanding that there is a causative gene, right? We know with FSHD, it's DUX4. I'm sure you've discussed it numerous times with experts and people who are working in the field. So it's been many years since discovery of the gene, and it was incremental understanding of how this gene is functioning. Why some muscles are more affected than others, why particular sequela in the disease. Mm-hmm. So it's therefore a straightforward thing to say, okay, because DUX4 should not be functioning in adult muscle tissue. While it's very important gene and protein in early development, it's in a fully functioning muscle, it should be silenced. So how do you silence that gene? The best approaches now are coming from understanding of nucleic acid therapy. Come in with what is called antisense oligonucleotide, right? A stretch of DNA or RNA that can bind to DUX4 RNA and silence it. So this kind of antisense oligonucleotide, or ASOs, is not a novel concept. There are successful drugs out there with this approach. So the question is really, how do we deliver this potent therapy, potent drug, or we call payload, to the muscle, to every muscle where it needs to go? And so this is really a solving of delivery issue for the drug we know should work. Okay, so that's really the task that on which DINE was formed. And so if you think about the muscle tissue in the human body, you think about 40% of body weight is muscle tissue. Okay. So it's a huge, we have also specializing muscles, right? Every muscle has a purpose. And for us to be able to deliver this payload to every muscle, you need to pick a technology to really get there. So this is where this component comes, not only oligonucleotide we need, but something else to take it to the muscle. So we created what is called force technology, to force the oligonucleotide into the muscle cell. And this is accomplished by an antibody. In fact, it's a fragment of antibody we call Fab. It homes to the muscle cell because it binds to muscle-specific receptor, okay? These receptors are highly enriched in muscle cell, in cardiac cell, smooth muscle, and so it can deliver the payload there. So to make this drug effective, we need to conjugate the two, link them, right? We link our oligonucleotide to the Fab through the linker. So here is our tri-component molecule. It has Fab, taking it to the muscle, linker, and actual drug or payload or oligonucleotide. Okay, so this is a unique approach that Dyne is developing. Okay, so that's so where we are. We would like to be able to just want to one more thing about our technology is overall our force conjugate is not only muscle specific, but it holds this intrinsic possibility to be redosable, so we can adjust the dose, right? Dose adjustable. We can give it every so often, depending on its functionality, and therefore it allows us to not only adjust the dose based on the need, but we can initiate it at a certain point. We can conclude it at a certain point. We can use it as potential add-on therapies to other type of therapies. So that's what kind of makes it interesting and unique.
You've said unique a couple of times within the explanation, and I can't help but think, does that equal then a lot of challenges because of the uniqueness of what you're trying to accomplish? And if so, what are those challenges?
Yeah. This is actually a very good question that you're asking about the uniqueness of it. Unique is in the way that it's constructed with the approach of picking the best way to home to the muscle, meaning the very potent part of the molecule, which is Fab. Another unique thing is how do we pick the best linker that would deliver payload? So we want to use the linker that can get to the cell, that is safe, and this is very effective and allows payload to be released once it's delivered to the muscle cell. So in this regard, it's unique. What makes it actually highly scalable and applicable is that once you do the work, those two pieces, which is Fab and linker, stay the same. And what is unique now is the payload. For FSHD people, we will use payload that is intended to silence DUX4 expression, right? But you can use the same force technology, the same Fab, the same linker, and just change the payload or actual drug to be able to target another muscle disease. To give you an example, where there's Duchenne muscular dystrophy, this is another disease we are working on, or it is myotonic dystrophy.
Yeah, myotonic, yeah.
So in fact, it's a plug and play technology that allows you to use the same warhead to deliver, and then actual drug will be unique to each indication.
So you find the right combination, then it can be copied or used again. Right. That's fascinating. Thank you for explaining that well, because sometimes the science can get over my head. I'm never ashamed to admit that. So I've learned a lot today, and you've added another nugget of knowledge for me. So I appreciate you taking the time to kind of, blow it up right for us and kind of diagram it out. And really, it does make a lot of sense. But like a lot of things, okay, there's challenges to it, but then there's some hope there to it for what you're saying, because, if you're onto that combination that can be repetitively used or payload, then how close are you? And Molly kind of alluded to this with certain quarters of, I think, next year, and then the first quarter of the following. So like 2021, 2022, kind of delivering some papers to the FDA. I mean, where are we along in the process? Then the side.
Right. This year has been a very productive year for us as we are trying to move very rapidly 3 programs. With FSHD specifically, we were able to generate the molecule that we intend to use. And we are what is called preclinical stage right now, or non-clinical, which means we are testing our therapy in model systems. Okay. And, as you may heard from others, we're using FSHD muscle-derived cells grown in the dish to kind of understand what is the behavior of our conjugate in the cells, Does it do what we intended to do? And so we are at this stage. And so it takes time to understand better how to dose, what is the effective dose that we need to deliver. And these are the necessary studies that are ongoing in this particular time frame. To what Molly has shared previously, we do this necessary preparation of what we call preclinical, non-clinical. We need to assess safety of the molecule and get all these pieces together in order to submit investigational new drug application at IND, towards which we are very rapidly progressing.
Mm. And since we're here, like on this, Giving Tuesday event, trying to raise awareness and funds, let's just be real what this day is about. Those two components, community involvement and funds, how crucial is that into your work and what Dine's trying to accomplish?
Right. It is very crucial on multiple fronts. We absolutely need engage. If you think about different companies approaching therapies from different angles, and they work in silos, that's not true. We all, the community, work together. So not only we work with you, with patients, we work with other people, we share our knowledge. We use advisors, key opinion leaders, and so we learn from one another. And it's a very healthy environment where we develop, it's a good thing to develop different approaches. It always gives us the choice to find this right ingredient, or right even combination sometimes of ingredients, and working together as community. So every step counts. In my view, I think that as fascinating as fundamental science is, and then you can say we are applied science. I can tell you, I see no difference. I see continuum from the early fundamental discoveries, using ideas that are ahead of us, and then as drug developer, we come in and bring our own dissection and knowledge to the field. And so it's only in collaboration that we can really go rapidly towards rational therapies, specific therapies to rare genetic diseases to benefit patients.
Thank you for explaining everything that Dine is doing, Oxana. I really appreciate the time today. I know our listeners do as well. Very excited to hear of what Dine's been doing, the hard work that you guys are putting into it. Young company or not, to me, that's okay. You're spearheading, and man, it's just really exciting times, and I'm sure glad that Dine is a part of it. And also being a sponsor of this event helps a great deal in the funding side of kind of keeping the wheel going here at the FSHD Society. And we really appreciate everything you guys are doing.
Thank you. Thank you, Tim.